If it's charity, yeah. They just obviously don't care, huh?
But it goes against us, though, right? Missing class and stuff.
So the good news about you taking my class is what am I doing right now? Recording. Yeah. What? Speaking of, why does this not look right? I guess I hit the wrong button earlier. Whew. All right, I started panic for a minute. This system like broke last semester, and I had to like go to a whole nother room. Okay, so we're coming up on the end. So as I said, next week I'm not going to be here on Monday for office hours, but I am going to be available on Friday in the afternoon for office hours, and probably in one of the 2 computer labs, either here or the Hibernia Center. I'll post an announcement for you guys once I check the schedule and make sure there isn't a class in there. Thursday is our test, right, on this last module, unit, whatever you want to call it. You have to take the test Friday morning at 11. If something happens to you on Thursday and you can't take it. So just keep that in mind because that afternoon I'm going to make the test available so people can review and meet with me if they need to. And then the following week is our final exam.
And it will be in the same building, that same place, right?
Yeah, our test is still in the Hibernia Center Thursday, right? No, next Thursday. Next Thursday, which is fast approaching, right? So we have one topic left after we finish up this topic of adaptive. And so I just posted the PowerPoint for you guys. I also posted a table that's in the PowerPoint, but I blanked it out. I find, right, you guys have been having me all semester long. You know me in tables, right? I find they're a really good way of organizing and collecting data and information. So what I suggest is that you fill out this table as you're going through, right, and looking at this material before lecture on Thursday. Okay? All right. So that's that's it. I'm not posting any more stuff, y'all. Yay. Right? The end is near. Okay, so I'll hit save. I wanted to do a quick poll, but let's see. I didn't type them ahead of time. But you know what? I can type on the fly. Right? So, which marker is, see who was paying attention last time. Do you want me to make it multiple choice?
Yes.
Okay. All right, so let's see. I gotta post it. It won't come up until I post it. Oh. Okay. Create. Okay, so let me send it. Okay, so which marker is found on helper T cells? Tyler, do me a favor, write a line under the last name if that happens to be yours. Okay. I deployed it, right? You guys getting it on your phones? You're just still thinking about it? does it not give me a number? I do not know. All right, anybody not agree with what has been picked? Okay. How many people are undecided?
Because it's not a button, it's fast.
Yeah. I think we had this trouble before, but the important thing too is just you guys seeing a question, right? Because I don't collect the data per se on you guys. The important thing is you guys thinking about this question for a second, right? Technology fails us, it's okay, right? So this is the correct answer, right? Yay. Bridget's excited. That's good. This is important, right, because this is actually one of the ways that we distinguish helper T cells from cytotoxic T cells. So what marker is on cytotoxic T cells? Which one on this list? CD8. And so remember, there's an important communication that happens between cells. So helper T cells talk to other cells, right? Who do they talk to? Anyone remember who helper T cells communicate with? Macrophages, yeah. That's one of them. They're in a group that we call antigen-presenting cells. So a macrophage is one of them. It'll take in stuff, right, break it up, and present those antigens on its surface. Who else does that? Dendritic cells, yep. They're major players in that. They're really important. That's one of their main functions, to share the information they gather, to present that antigen. One more. MHC class 2 is the marker that's on dendritic cells and macrophages, and one more cell that's an antigen-presenting cell that will actually take in antigen and present it to the helper T cell to get help from that helper T cell. Who may need help from the helper T cell? They make antibodies. That's a big clue. What cells make antibodies? It is a lymphocyte. You're close. You're getting warmer. B lymphocytes. A T cell is not going to talk to itself. Right? So T lymphocytes, right? These are really important interactions, y'all. So CD4 and MHC class 2, remember these go together. We sing the little song to ourselves, 2, 4, 6, who do we appreciate, right? And CD8 communicate with MHC class 1, which is found on all of our nucleated cells, including those antigen-presenting cells, the macrophages, the B cells, the T cells. So those are important components for our adaptive immune response, this communication that happens. And so, I'm trying to remember.
It was right before colonial selection of lymphocytes.
Yeah. So I know we looked at clonal selection last time. I think that's probably where we ended, right? Is a clonal selection of B cells. Yeah, I'm pretty sure it is. Okay, so there's one thing that I wanted to show you guys that's on our Canvas site. So remember, on our videos and animations, right, we have all the videos and all the animations I've showed you guys, or will show you, or will tell you to refer to. There's one that I added right here. It's a YouTube link. It's on T-cell development. So as we just mentioned, and there's a reason why I mentioned this, right, that communication is really important, of T cells being able to communicate with the different MHCs. So remember, T cells are named after what organ do they learn this distinction and become helper T cells and cytotoxic T cells? In the thymus, right? That's why they're called T cells, right? This education happens in the thymus. Where do all our blood cells originally come from, though? From the bone marrow. So we call those our primary lymphoid organs, right? The T cells are educated in the thymus, so that's one of our primary lymphoid organs, and so is the bone marrow. So this is going to happen in the thymus. So hopefully I don't have to deal with.
T-cell development takes place within the thymus, a specialized organ where T-cell progenitors mature and differentiate. The thymus is divided into several different anatomical locations, and as thymocytes mature, they pass from one environment to another. T-cell progenitors enter the thymus through high endothelial venules and migrate to the subcapsular region, where these thymocytes start their process of maturation by rearrangement of their T-cell receptor genes. As maturation proceeds and the cells begin to express a specific receptor, they move further into the thymic cortex. Interactions between the T-cell receptor and MHC peptide complexes displayed by the thymic cortical epithelial cells play an important part in the fate of the thymocyte. At this point, the developing thymocyte expresses both CD4 and CD8, as well as an alpha-beta T-cell receptor. A cell that has a receptor able to recognize an MHC class I molecule receives both a survival signal and a maturation signal. Eventually, the cell stops expressing CD4 and maintains expression of CD8. Thymocytes that obtain a survival signal are said to undergo positive selection. A cell that has a receptor able to recognize an MHC class II molecule also receives a survival signal and a different maturation signal. Eventually, this cell stops expressing CD8 and maintains expression of CD4. Thymocytes whose receptors are unable to recognize either MHC class I or MHC class II molecules fail to receive any survival signals and die by programmed cell death or apoptosis. Thymocytes that are able to recognize MHC class I or MHC class II peptide complexes too avidly receive a strong signal that drives them into cell death. In this way, thymocytes capable of responding to self-peptide antigens are eliminated in a process known as negative selection or central tolerance. The surviving T cells migrate from the thymic cortex to the medulla. At this stage, they remain capable of recognizing self-peptide antigens expressed on other cell types, such as dendritic cells or thymic macrophages, and receive a sufficient signal to cause them to undergo programmed cell death. The remaining thymocytes, now mature, naive CD4 and CD8 T cells, pass out of the thymus, either returning to the bloodstream directly, passing into venules, or via the lymphatic system.
Right, so that very important, like I like to refer to it as, let me make sure that I...
T-cell development takes place within the
there. That really special kind of handshake you could almost think of, right, that needs to happen between our cells. So that education of which you're going to be, are you going to be a helper T cell or are you going to be a cytotoxic T cell, happens in the thymus. And as you saw, they test it out, right? Helper T cells have to be able to recognize MHC class II. And remember, they're helping out, right? So they're communicating with specialized group of antigen-presenting cells, which we mentioned earlier, are which three cells? Dendritic, macrophage, and B cells, right? Where CD8, cytotoxic T cells, can communicate with MHC class I, which is found on all our nucleated cells. So all our cells, right? So including dendritic cells, including macrophages, including B cells, right? B cells and macrophage and dendritic cells are just special and that they have MHC class II as well. So they can talk to the helper T cells. And of course, one of the key things of our adaptive immune response is this ability to tell the difference between self and non-self, right? We don't want T cells that recognize self. Are we locked again today? Do I need to call and get our door? Is locked? Ugh. Yeah, don't close it all the way then. Delosier isn't as loud as she was. She must have closed her door. So, or Strickland, sorry. Old last name. Yeah. We all have distinctive voices. She's across the hall. So I just couldn't compete with her earlier.
I have a question. So, like, if the thymus, say for instance,
Mm-hmm.
For some reason, we have a patient that has received several vaccines, but when we do a titer on one particular titer is not showing up in it.
Uh-huh.
They don't have any immunity to it. So
Right.
where's that from? I thought we artificially placing it in them.
Right. Yeah, so good question. Actually, Bridget asked a really good question. So they have a patient that has been vaccinated against a particular disease. And when you do a titer, and what you're doing when you do a titer is you're taking a blood sample and you're seeing how many antibodies somebody has against that particular vaccine. For this particular person, they're not making antibodies. So it could be several different things that could be going wrong, right? That might be a red flag. Hopefully it's just to that one, not to anything else.
Well, I can concur with them because for some reason,
Yeah.
it's MMR. I took the vaccine the same time with them
Uh-huh.
to see if it was a flu.
Yeah.
For months, we're not any immunity.
No immunity. Okay. But to the other things, you're immune. Okay.
So it's kind of crazy. It's like,
Right, yeah. So the reason why someone might not develop antibodies is that your body is not recognizing the vaccine. Yeah, and that's the unfortunate thing. That's why you're not producing antibodies. Now, it could be the different levels. Think about how does an antibody come about, right? So your body takes it in, right? And it's looking for, like they said, naive cells. Naive meaning they've never seen it before. But this is a great lead into our next thing is how is it that we make different T-cell receptors and antibodies? Like, how is it that we actually make them? And that's the next thing that we're going to talk about. So the question is, somewhere in the line, it's not working. Yeah, that's not good.
that posed a problem for me then because I'm working in healthcare and she was like, you need to get another MMR. And I'm like, I need to see 12.
Yeah, yeah. I don't know what they'll do for that. I would say the unfortunate thing is that then you're at risk of potentially getting that, and if they ever have a breakout, then you can't be involved in helping. You know what I'm saying? Because you're not protected. Yeah, it's not good. Not for measles, not good.
It's mumps.
Oh, mumps. Oh, mumps is not usually that big of a deal.
Rubella and measles is the
Yeah.
mumps.
Yeah. And remember, we talked about this one earlier too. Oh no, it's my other class. That vaccine isn't very effective. I think it's only around 60% too.
Okay, 'cause I was about to
So,
say,
yeah, that I guess the issue is that maybe they need to do some work on that vaccine because a lot of people are running into that same problem you are, where like the immune system isn't really. So the question is, are we priming the immune system enough to get people to make antibodies against that virus? So let me get out of this and go back to our PowerPoint for today. So once you have all these B cells and T cells, and they're continually being made, right? Each one of them is unique. So, for instance, for a B cell or a T cell, where they show a B cell in this diagram, it makes its own unique antibody. For a T cell, it has its own unique T cell receptor that recognizes a specific antigen. These cells, when they haven't encountered antigen, are referred to as being naive, right? Just like a little kid, right? You're naive to the fact that there are bad guys in this world, right? Until your parents teach you not to talk to strangers, right? You don't really know, right? And you definitely don't want to find that out the hard way, where unfortunately some people do. So here we have naive cells. Once they come into contact with antigen, they can become activated. Once they activate, you don't just want one B cell that recognizes that pathogen, right? You want multiple ones. So in activation of lymphocytes, you always get proliferation, where they're going to multiply, right? So instead of just having this one B cell that recognizes the antigen, you're going to produce millions of them, right? All clones of themselves, right? They're all replicas. They're all very similar. They all arose from the same cell. That's why they call it clonal expansion, right? Clones meaning a copy. Some of them, as we know, are going to turn into plasma cells and actually secrete the antibodies. Right? So those proteins are going to actually secrete them. We know how awesome antibodies are. I don't need to go over that. And then always, whenever lymphocytes get activated, whether it's a B cell or T cell, you're going to get memory cells produced. And so if you see this antigen again, those are what are going to be activated probably, as opposed to having to try and find naive cells. Right? So, again, activation is going to be proliferation. They're going to have interacted with their specific antigen. Effector lymphocytes are ones that are going to actually do something. So in the case of plasma cells, they're the ones that actually make antibodies and secrete them, right? What's wrong?
I don't have that. I'm not missing.
Oh, okay. T cells can either be helper T cells, so again, they're going to help out those other cells, B cells, macrophages. They're usually activated by dendritic cells, though. Cytotoxic T cells, right, are going to go and kill infected cells. And then always, as I said, memory cells. And these are much longer lived descendants of those activated lymphocytes, those T cells and the B cells. And they're the reason why our secondary response, the second time we see that specific antigen, our response is much swifter, much faster. So, here, right, the very first time you see an antigen, usually you're going to have a lag period before antibodies are produced. It says 10 to 12 days here. The general average is 7 to 14 days. So say you get vaccinated, right? They're not going to take your titer tomorrow to see if you produced antibodies. When are they going to take it? Yeah, they're going to actually usually wait about a month. Right, to check your titer. That lag period is when they're trying to find a naive T cells and naive B cells that recognize that specific antigen you've been vaccinated against or that you've been exposed to. That's the scary thing about this, is that there is a lag time. There is a time when you're not protected by antibodies because you have to first make them. You first have to find and activate naive cells, lymphocytes, that recognize that antigen, if it's the very first time you've seen it. The good news is that our system, right, has the ability to remember. So we take advantage of this by vaccinating, so that the first time you're exposed in a way that shouldn't make you sick, right? And yet we're going to get an immunological response. We're going to get you to produce antibodies, or maybe cytotoxic T cells, or both. Right? Most of the vaccines that we give you, you're going to produce antibodies against the pathogen. So that way, if you see it again, you're not activating naive cells, you're activating what? Memory cells. And it's much faster reaction. No lag time. Yeah, that's what you want. That's what you want. That's why we vaccinate. We take advantage of this ability of our adaptive immune system to remember, right? To produce memory cells. What has to save your butt in the meantime between getting infected, either naturally or vaccinated, especially if you get infected naturally? What are you waiting on? You're waiting on an antibody production or a cellular response. What has to keep the bacteria down or the virus under control?
Inflammation or older antibodies that are still active.
All right, you can't have older antibodies the first time you've ever seen it, right? So you're right about inflammation, because what does inflammation bring to the area? White blood cells, right? That's the whole purpose of it, is to bring the white blood cells there. And what type of white blood cells are there? Are being brought there?
Macrophages.
Macrophages. and neutrophils. And what are their main functions? To eat, phagocytosis, right? So your innate immune response, right? Your inflammatory response, those phagocytic cells, those antimicrobial peptides, complement system, all of that is going to try and keep it under control, right? If it eliminates it, great, right? But if it can't, that gives time for the adaptive immune response to, say, produce antibodies or cytotoxic T cells that will go into the area and preferentially target and eliminate those invaders. Make sense? Right? For some infections, unfortunately, that we vaccinate against now, your immune system wouldn't probably produce an antibody in enough time to save you, like tetanus, because it's a toxin that gets you, right? And it can spread system-wide. You don't have 7 to 14 days, right? You've got maybe a day, and then you're going to stop breathing and die, right, without life support. How are they going to treat you with that? If you have tetanus, right, and you don't have enough antibodies, you haven't been properly vaccinated.
Tetanus shot?
Well, they're going to give you that so that in the future you'll produce antibodies. They're going to give you somebody's antibodies, someone else's antibodies. They're going to give you what's called antitoxin or anti-serum. They're going to give you someone else's antibodies to help protect you until you can make your own. And depending on when they catch it, right, they might have to put you on life support until the toxin gets cleared from the system. And the good news is that the damage that it causes to the interference it does for the inhibitory motor neurons is not permanent. So it can be relieved. It's just you kind of have to breathe in the meantime, and there's muscles that help you do that, right? And they need to relax and then contract and then relax again. They stop relaxing with this toxin. Does that make sense to you guys? Right? So vaccination is really paramount to this ability that we have. I think I mentioned this earlier about the rough endoplasmic reticulum for plasma cells, right? Gets huge because, remember, antibodies are what type of molecules? Are they DNA? Are they protein? Are they fats? Are they sugars? They're proteins, right? And what's on the rough ER that makes it rough? Ribosomes, which make proteins, right? And that's why plasma cells, their rough endoplasmic reticulum gets huge because they're going to secrete those antibodies. So, antibody diversity is going to be there. Okay. So, with antibodies, we want them to bind to that epitope, to that little piece of the antigen, as strongly as possible. So when B cells are activated, they actually purposely, spontaneously mutate as they multiply. So they're not actually going to be 100% exact copies of each other. They go through these little tweaks in their DNA to change it so that they change their antibody. But again, this is random, and what they're trying to do is to create an antibody that binds much more strongly to the antigen, to that epitope on the antigen each time. So because this is random though, right, what are the chances that it's going to bind better? It's random, right? It's possible, but you could also end up with some that don't bind as well. So right here, what they're showing you with the pluses is showing you the level of binding. So say right here, here's this B cell, it's been activated, it recognizes a specific epitope on an antigen. When it divides, it mutates its DNA. One of the mutations that it does, this spur off of it, they end up being less binding, right? So 2 pluses means it's binding 2 times, right? Our 2 level of strong. Where in this case, it becomes less strong of a binding because of the change it made. Is that B cell going to get a signal to continue on and keep making copies of itself? No, because this original one was binding much better, right? So that good binding is going to continue that lineage down, keep getting signals to proliferate. The next round, say they actually end up with one that actually has a better binding, a 3+. That one's better than the 2+, so this one is going to predominate. So as they go, like I said, they're continually changing and mutating their DNA in hopes that they get a better affinity. This is referred to as affinity maturation. The other thing that happens when you have T cell involvement is you can have what's called class switching. So remember, we're programmed, the very first antibody we produce is IgM, right? That mega big one. We're not going to switch to any of the other classes unless T cells get involved, if helper T cells communicate with the B cell. And this is a change in the DNA again. And we're going to watch a video in a moment that shows you how B cells make antibodies, how they change their DNA to make their own unique antibody. And then I have a little drawing I'm going to do on the board. So I just figured out how to explain one of the things I want to explain. At least I think I have. So this switching, remember, where would we want to do this? Especially the MALT. What does MALT stand for? Close, it's the other M. What are all our openings bathed in? Yeah, mucus-associated lymphoid tissue. So remember, in our mucus, we make IgA, right? That's our secreted antibody that goes into our secretions, into our mucus, our breast milk, our tears, our saliva. So it makes sense that in the B cells in the mucus-associated lymphoid tissue, they're going to be asked by the helper T cells in that area, say, hey, you need to make IgA, right? And in the lymph nodes, they're going to make IgG. In allergic individuals like myself, they're going to make IgE, unfortunately, right? So I already mentioned this, right? Vaccination exploits this memory, right? It allows us to jump right to secondary response, hopefully, if we're ever exposed to something we've been vaccinated against. And we don't have to worry about this lag period. So how is it that we make different antibodies? And although we talk about antibodies here, the same thing happens for the T cells for their T cell receptors, right? So each T cell recognizes a specific antigen based on its T cell receptor. Each B cell recognizes a different antigen based on its antibody, its B cell receptor it makes. So what it does is it does gene rearrangement, right, where it literally rearranges the genes, the DNA, right, the code on how to make these receptors in their DNA. It does it imprecisely, right? When it picks out the different segments, it doesn't do it precisely. And this is the one thing that they don't show you in the animation that I'm going to try and show you by doing a drawing on the board. Okay? Combinational associations. So when we combine things, right, we create more diversity. So those two things, the rearrangement are shown and the combinational associations are shown in this animation. All right, so we're gonna watch this, talk about it, and then I'm gonna also show you the third thing, the imprecise joining. So.
Human cells do not have enough DNA to have separate genes for each antibody molecule. Instead, different segments of DNA can be mixed and matched to form different antibodies. Light chains are made from V and J segments in the variable region and a constant region segment, whereas heavy chains are made from V, D, and J segments in the variable region and one of 5 different constant region segments. As the lymphocyte divides during maturation, its genes are rearranged. DNA sequences are assembled by the random selection of V and J gene segments for the light chains and V, D, and J segments for the heavy chains. The constant region of the molecule is encoded by a single C or constant gene segment for light chains and 5 different constant regions for heavy chains. The rearranged DNA in the mature B cell is transcribed into messenger RNA and translated into the light chain of the antibody. DNA sequences for the variable regions of each heavy chain are assembled by the random selection of a V gene segment, a D gene segment, and a J gene segment. The genes for the constant regions of heavy H chains have Greek letters designating IgG, gamma, IgM, mu, IgA, alpha, IgE, epsilon, and IgD, delta. The DNA in the mature B cell is transcribed and translated into the heavy chain protein. The light chains are then attached to the heavy chains to form the functional antibody. Because of the large number of ways in which the genes can be rearranged, many different antibodies can be produced.
So I always equate this to basically B cells and T cells pick their lottery numbers, right? Because they're literally picking segments of the DNA, right? And so they have lots of choices, just like when you go to play the lottery, right? You have a bunch of choices between numbers. They have a bunch of choices of segments of genes on how to make this protein. And they pick their numbers, right? So in this case, they picked V3 and D1 and J2. That's for the heavy chain, right? And then for the light chain, they also had to pick a V segment. In this case, they picked number 2 and J number 1. Do you see that? Right? So that combination, right, that rearrangement, picking different ones, picking which V, which J, that's rearrangement. Because they literally pick out and they throw away the other pieces. So just like when you hand in your lottery ticket, that's it, right? You can't go, I want to change my numbers, right? It doesn't work that way. Same thing for these guys. Once they pick their numbers, that's it. It's a permanent change in the DNA, right? They change their DNA. Does that make sense to you guys? So where do we get a combinational diversity? Well, what make up antibodies? Heavy chains and light chains. So notice that for the heavy chain, right, we have the segments we picked, the V's, the D's, and the J's. That's all variable. And for the light chains, you had the V and J segments that they chose. It is the combination of both the heavy and the light chains that make up the antigen binding site. So this is that combinational diversity. Still with me? Okay. So the other thing is imprecise, and I'll try and do a PowerPoint drawing and post it up later. I've been thinking about this one for a while. So for a particular B cell or T cell, right? Imagine you have a gene segment that I have labeled 1, 2, 3, and 4. But these are gene segments, right? So this is, we're talking about DNA, right? So this is A, T, C, G, A, T, T, G, C, T, A, T, T, C, A. I'm just randomly making up a code, by the way. Okay? That's the code, right? So for this particular segment, that's the code of DNA. Now, do you think when they choose number 1, they take from A to T right here? I guess I need a different color. I take the whole thing?
No.
No, because that would be precise, right? We would precisely take all of 1 and say put all of 1 with all of 3. We could do that, right? But that's not what it does, right? It does this imprecisely. So it will say take this section of 1 and combine it with this section of 3. And put those two together. That's imprecise, right? So even if another B cell or T cell picked 1 and 3, because it does it imprecisely, doesn't take the whole 1 and 3, you're still going to get variability, right? Because the next B cell may pick, I need a brighter color, they may pick this section of 1. They're going to pick this, and they're going to pick this section of 3. So even though they both picked the same gene section or region, it didn't take the same piece. That makes sense to you guys? So we have 3 levels of variability, right? We're going to pick segments of the gene. We're going to do that imprecisely. And then for antibodies themselves, you have the combination of light chain and heavy chain that make this antigen binding site. So 3 levels of diversity in forming an antibody. Or even a T cell receptor, right? Because T cell receptors have two proteins in an antigen binding site. This is random though, right? It's randomly choosing its numbers. It's randomly putting them together. And you make millions upon billions of these. I don't want to lose my mark. Those are stolen from my son even, so. He doesn't use them. I buy him tons of markers. No, it was just an example. Yeah, that's what I'm saying. Each B cell, as you see here, right? In this case, it picked 100, 4, and 6. Right? It's random. Right? It's like when you go play the lottery and you say, give me a quick pick. It's random, right? There's a chance you could win. So for these guys, when do they win? What's winning for them?
Being copied and drawn to the next section of the cell.
Well, each B cell makes its own unique antibody, right? At this point, when it comes out of the bone marrow, right, it's hoping it runs into what?
Upper T cells?
Whatever it's supposed to bind to, right? Whatever antigen, whatever epitope, right? For them, that's winning the lottery. And for you, it is too, right? Because if that B cell recognizes an antigen, it's going to make antibodies against it, right? And we already know how awesome those are. Right? So for them, right, we make tons and tons of naive B cells. But this is why it takes time, right? Because we've got to find that naive B cell that recognizes that antigen. Now the good news is the antigens are quite complex, right? What's the chances we're only going to make one antibody? Pretty slim. We'll actually probably make several antibodies against an antigen. Because remember, antibodies can bind to specific sites on that antigen, specific epitopes. The molecular interaction here is really small. It's really specific, though. So that's kind of scary, huh? Because I don't know about you, but I still haven't won the lottery yet. Granted, I don't play as often as other people. But, you know, our B cells win the lottery all the time for us, right? That's because, we have millions of them, right? You're not just buying one lottery ticket, right? You're buying millions of lottery tickets all the time. Still with me? Right? Hence why, thank goodness, as a collection, right, our B cells win quite often, right? Our immune system wins quite often. There's something I wanted to say. Okay, so why don't we just have DNA? Why don't we just have DNA that makes an antibody against, say, mumps? Why aren't we programmed? Like, why didn't my mom give me the DNA to make an antibody against mumps? Come on, Mom. Come on, Mom, right? She should have helped you out. Or your dad, for that matter. You get DNA from him, too.
Maybe they never had mumps.
Right. But think about this, though. Is mumps going to stay the same forever?
No.
It might change, right? The influenza virus. Why do we get vaccinated every year? It changes. It changes. So even if, say, we had the ability to pass on to our children how to make the antibodies that we've made, that would be a lot of DNA for one. Would that really protect them? No, because pathogens do what? They change. So if we, too, do not have the ability to change, then we wouldn't be able to keep up with it. So this system isn't perfect, but it does give us the ability to continue to change, because if we don't change, the other organisms we're going to get outcompeted. So I have to say, Friday, I think I was probably even complaining to you guys that I had to go to Baton Rouge. I'm actually really glad that I went, because I found out about a project that I want us to start doing at Delgado. There's a microbiology project called Small World Initiative. And in microbiology labs, instead of doing an unknown, which I know and I give you a bacteria, but I know what it is, we're actually going to do real unknowns. Like take bacteria from the soil and try to identify new bacteria that produce antibiotics. Pretty cool stuff. Huh? Yeah, I think that I don't know. It's going to be a lot of work, but I think it'll be awesome, right? Because we'll have the potential of discovering new stuff, right? And that's the thing is that a lot of science is like that too. Like I have a friend at BRCC, she's already doing this program with her students. And a lot of times, they get bacteria that we already know about. Or they get ones that don't produce antibiotics. And research is like that, right? There's a lot of, we didn't get what we wanted. Let's try something else. So that's kind of like what's going on in your body too, right? Is they just keep trying different ways. Keep trying until you find one that works. The scary thing about that is it can take a long time, right? Still with me? All right. So, we talked about that. All right, so I think this will be a good one to go through. So let's watch this one.
Proteins generally require the cooperation of T helper cells, Th2, to stimulate B cells into becoming antibody-producing cells and memory cells. Such antigens are therefore said to be T-cell dependent. The protein antigen is first ingested and then broken into peptide fragments by an antigen-presenting cell, such as a macrophage. The antigen is then moved to the surface of the macrophage on a class II MHC. The T-cell receptor on the surface of a T helper cell recognizes the peptide antigen that is being presented on a class II MHC of the macrophage, and the T helper cell becomes activated. The activated Th2 cell is now capable of stimulating B cells. B cells also ingest antigens. The antigen reacts with an antibody on the surface of the B cell and is then internalized, digested, and presented at the surface of the B cell on a class II MHC. The antigen class II MHC complex of the B cell interacts with the T cell receptor on the activated T helper Th2 cell. The T helper cell produces cytokines, which stimulate the B cell to proliferate and differentiate into antibody-producing plasma cells and memory cells.
Okay. Right? So macrophage, right? Who else could this be other than a macrophage that ingests something and presents it? Dendritic cell, right? So it engulfs it, it presented it. What molecule presents it? MHC class 2, so therefore he's going to communicate with helper cell. And so notice here, this is one thing I like about this one, right? So there's the T cell receptor. It's recognizing the antigen, right? The MHC class 2 is presenting. What's this molecule? What makes this a helper T cell? This is CD4, right? This is how this cell knows to communicate with this cell, that it can communicate with MHC class 2. Still with me? Right? So it recognizes it, right? So these guys communicate. How do they communicate? What are their voices that they use to talk to each other? It's a general name we give to these molecules. The hormones of the immune system. Very important, definitely on the test. How do the cells communicate with each other? What molecules do they use? What's the general name we give to those molecules? Starts with a C. Cytokines. Cytokines, right? Cytokines. Right? And then this helper T cell has been activated. So is it actually going to be just one of these cells? No, what are they not showing you here? Do you just have one helper T cell, right? It's been activated. So one cell becomes 2, 4, 6, 8, keeps on going, right? They're going to proliferate, right? All lymphocytes, once activated, proliferate. So you don't just have one helper T cell now that recognizes, you have thousands of them. Right? So then that T cell can go and communicate with this B cell, which has already encountered antigen, right? And is displaying it on his surface. And what molecule? Ooh! It went really fast on me. What molecule is that? How do antigen presenting cells present antigen? MHC class 2, which is going to allow it to do what? Talk to this guy, right? This helper T cell. Because again, MHC class 2 can communicate with CD4. And in this case now, this helper T cell is going to do what? It's going to activate that B cell. So we're going to have just one B cell? No, they're going to sing along with me. They're going to proliferate. They're going to multiply, right? And B cells, when they proliferate, they're also going to then differentiate into two different types of cells.
Plasma cells?
Yep, plasma cells and?
Other B cells?
Nope. Who's going to help us out the second time around? Memory cells. And again, another thing that they didn't show you is going to get helper T cells that are memory cells as well, right? That are going to stick around. So here, right, we get proliferation and differentiation. Some plasma cells, right, some memory cells. So that's if T cells help out the B cell. And if that happens, you can produce other classes of antibodies like IgG or IgA or IgE or IgD. T-independent antigens, so these are antigens that tend to be repetitive, right? Like polysaccharides that contain a lot of the same sugars. Because of that, they can interact with a lot of antibodies on the surface of a B cell. If that happens, that usually could be sufficient enough to activate that B cell without help from the T cell, hence it's called T-independent. And in that case, though, there's only one class of antibody we produce. Which one? What's the first one, the only one produced by the fetus, doesn't leave the bloodstream, is huge when we produce it? It's mega big. IgM. IgM. So we've gone over that. Most of this stuff is kind of repetitive. I want to go right here. So this is a drawing from that first website I sent you guys to, right? And it's cute, and it's a really good overview, as that website was. But now we can look at this and we could add in some details, right? From what we've learned. So we know a B cell has a unique B cell receptor and antibody. They didn't draw it really that good because they're only showing one antigen binding site. It doesn't look like an antibody, right? It's not Y-shaped. There's two reasons why I know this is a B cell, other than the fact that they labeled it, is because it's directly interacting with antigen. Only B cells can do that. So they say this is a little bacteria, right? And his little epitope right there, right? That's what the little crown on his head is the epitope, right? That specific portion of the antigen of that bacteria where the antibody is binding. Now, one of the things that may need to happen, because again, this bacteria is only binding to one antibody, right, on the surface. That's not sufficient to cause this one to proliferate. So this B cell probably is going to need help from who to be activated?
T cell.
T cell. What T cell?
T helper.
The T helper cell, which we know is CD4, right? So again, they show this interaction here, but they're not showing the B cell presenting the antigen, right, or the T cell recognizing that antigen. This is something that would maybe help you, right? And sometimes I used to tell my students, draw this stuff out, right? Draw these interactions out. It will help you. And they didn't have any clue as to what I was talking about, right? So I had done a special lecture a couple semesters ago, and actually had one of my students record me writing and drawing on the board. So what I'm going to do for you guys is post that, and what I want you to do is I want you to also draw out this stuff and follow along in the video. Okay? I don't care, I don't care how pretty your drawings are, right? It's the act of doing it, right? It does not have to look pretty. Mine are not pretty, right? I am not a good drawer. It is part of the active learning process, right? And I'm so passionate about it that I'll let you guys take pictures of your notes, right? And upload them for bonus points in Canvas, okay? So I'm going to be turning that assignment on for you. It's 4 separate videos of me going over these types of interactions, right? Going through the process and drawing it out, as well as with antibodies, right? Because again, I feel very strongly about it, that if you guys actually spend the time and draw it out, you'll really learn it. So what happens next? So B cell communicates with T cell, gets activated. B cell does what? It's activated. What's next? It divides, right? It proliferates. And then some of them are going to become plasma cells and secrete antibodies. Others are going to become memory cells to stick around in case we see this again. So see, not very complicated, right? Kind of cute, right? It's one of my favorite ones, right? Because the plasma cell literally spits out the antibodies. right? Sometimes these visuals help us, right? Okay. So then those antibodies, we know, right, can bind to the pathogen. So what's one of the antibody outcomes that will happen, especially if you have a bacteria and the antibody binds to it? What could potentially happen to that bacteria? It could be eaten, right? Because phagocytic cells have receptors for antibodies, don't they? For the Fc portion, right? This end of the antibody. What is that type of antibody outcome called? We have a special name for it. When antibodies bind to the surface and tag it for destruction by phagocytic cells. Yep, opsonization. Opsonization. So right, here's a macrophage, right? Or a dendritic cell, I'm not sure which. Right? He's like, woohoo, this one looks delicious. Right? Because it's coated in antibody, right? It says that this is definitely a bad guy, right? Our immune system is recognizing it as foreign. Is that the end of the story? We're going to gobble all these guys up and the bacteria are going to be gone forever and ever? We could only be so lucky, right? Is the possibility this invader may invade again? Yes. So who are we going to have that's going to help us if this happens again? The memory cells, right? He's going to be like, oh, I remember that one. The plasma cells actually secrete the antibodies, right? So remember, antibodies can be extracellular, right? They're secreted. We want that, right? We want it to go into our secretions. We want antibodies all throughout our blood and our body tissues. Because this is only one of the outcomes, right? One of the things that I'm going to go over in that video is review all the 6 protective potential outcomes of antibody-antigen interactions. This is just one of them, right? This is just one of the possibilities of how antibodies can help us. So the other good thing about this diagram from your book, or it might be from another book, I steal stuff sometimes, is it kind of shows you where all the possible outcomes and interactions that can happen. Remember when we talked about leprosy, all of this could potentially happen, or, right, sometimes we kind of go all to one side, right? And we don't really have the ability right now for some infections. So maybe, Bridget, maybe your immune system is going the cellular route, right? Maybe you have cytotoxic T cells that recognize that virus. We don't really have the capability to test for that. We do have the capability, it's really easy to test for antibodies present in our blood, right? And for most things that we're going to be vaccinated against, even viruses, antibodies are helpful, right?
Yeah. So is it good, is it safe to say if you even saying you equivocal? I think that was what it said, equivocal. Like that was equivocal, like had some antibodies.
Oh, so you have some antibodies, just not a lot.
Like it was one away from the I guess the
Oh, okay. Well, that's good. That's good. That's better news.
But
That's
he
better
wasn't.
news.
That's because it was like I was the only one willing to take it because it was like a parent is getting upset because they have, since he was 6, he has received 5 of he's 18 trying to get into college. The college just won't take the vaccine record. Wants that. Yeah.
Wants tighter.
She has to keep spending money on the titers, and that titer is not cheap.
Yeah, no, it's not. That's ridiculous. Yeah, there has to be something with that. But yeah, that's crazy. That's crazy. So think about this. So think about, right, and again, almost tell yourself little stories, right, as we talk about what potentially could or should happen as far as an immunological response. So imagine, right, you get a cut and bacteria gets into your tissue. And in the inflammatory response, as that fluid leaks into that tissue, that fluid is picked up and goes through your lymphatic vessels. Your lymphatic vessels, right, your lymph, that fluid in it, is drained before you put that back into circulation. What is it drained by? What is your lymph drained or filtered by? What filters your lymph? lymph nodes, right? Your lymph nodes, that's what they're named after, right? The fact that they're nodes, they're sections, right? But they're where filtration of lymph happens. So that would be an example, right? So here's your primary lymphoid organ, so your bone marrow where your B cells are born, and your T cells, but remember the T cells have also got to travel to the thymus. Those immature T cells and B cells, those naive cells, right, so once they've matured here, travel out to the secondary lymphoid organ. So they're in the lymph nodes, right? They're in your mucus-associated lymphoid tissue, your skin-associated lymphoid tissue. They're in your tonsils, right? They're all throughout your body. They're in your spleen. What is your spleen filter? Blood, right? Filters your blood. So here are those naive cells, right? Naive cytotoxic T cells, naive helper T cells, CD4, and naive B cells. Now, say this is a lymph node. One of the things that could happen, too, is that lymph may contain dendritic cells from that tissue even that have already gobbled up the pathogen. So they'll travel in the lymph to the lymph node, and they'll be able to present that information to the T cells, whether it be helper T cells or cytotoxic T cells or even both. That presentation, right, dendritic cells are really important in presenting to our T cells because our T cells have to have antigen presented to them by a cell. The other one that can sometimes do this, anyone know, that can present to a T cell to activate it other than a dendritic cell? Who else is on the scene all the time guarding entry? So you have dendritic cells and who else? You got it. Starts with an M. Macrophages. Macrophages. The big eaters do a lot. Yes, the big eaters do a lot. They do a lot more than just eat. But your dendritic cells are really the main ones that usually do this antigen presentation, but macrophages can do it too. Right? I don't want you guys to forget that. So once that happens, right, once he presents, these cells will proliferate. Right? And as I said, and one of the good things that they have here is, look, memory cytotoxic T cells, memory helper T cells. Right? If they get activated, some of them are going to stay, say, in that lymph node as memory cells. Right? Or in the spleen or the tonsils. Right? Wherever that entry point was, that contact point. The effector cells, the helper T cells, can travel out from that location. And especially for cytotoxic T cells, you may have virus infection at that site where that dendritic cell came from. You want those cytotoxic T cells to then travel, right, through the blood, get to that site of inflammation, and attack those virus-infected cells and destroy them. The helper T cells may stay within, say, the lymph node and help out B cells that also encounter antigen that was brought along in that lymph, into that lymph node. B cells, remember, once they're activated, and they may need help from the T cell in order to do that, and especially if we need to change to a different class of antibody, right? We don't want to just stay at IgM. What if we need IgG? If in a lymph node, that's what most of them are going to make. Those will proliferate, and again, we're going to get memory cells. Those guys are going to stay put. The plasma cells themselves can either stay in the lymph node, and some of them can even travel to the area where the infection is, and they'll make antibodies right on the scene. These are effector T cells, whether they be helper or cytotoxic or plasma cells. Once we've cleared the infection, these cells are going to go through a process called apoptosis, which is programmed cell death without inflammation. Right? When our cells are naturally told to die, right, we don't want inflammation. That process is called apoptosis. We're going to kill off these cells because cells are expensive to maintain, right? Just like an army, right? We always have this debate on how much armed forces we really need because it's not cheap, right? Nor are maintaining cells that you don't need. Why can we afford to get rid of these guys? What do we have as our backup in case we see this bad guy again? We got the memory cells, right? So once those guys get activated, right, we're going to produce more plasma cells, right? Or active T helper cells or T cytotoxic cells. One of the other things that helper T cells help out with the macrophage's killing ability, right? It can make them more powerful killers. So you see why I love this diagram, right? It has pretty much everything in it. But again, you got to kind of tell yourself a story as to what's happening here or what could be happening. Right? So definitely, like I said at the beginning of class, for next time as you start reviewing, because we're going to start the last topic on Thursday. We just got a few things left in this one that I need to talk about with you guys, right? This natural killer outcomes. All the rest of this we've pretty much covered, right? And that bonus assignment's really going to help you guys, right? So, I got to look it up.